Hi, I'm Chris Contag, and I'm the director of the Institute for Quantitative Health Science and Engineering at Michigan State University. I got involved in orthopedic research a number of years ago when I was still at Stanford University. And we'd created a number of new imaging techniques that would allow you to watch biology in vivo. We oftentimes talk about making animal models transparent to scientific investigation. This included bioluminescence imaging and fluorescence imaging. And we were applying these imaging tools to tissue engineering and began to focus on bone regeneration in critical defects and distraction osteogenesis with my colleagues in orthopedics at Stanford. And then with great interest, we really contemplated new materials as a fundamental potential mechanism to target better bone regeneration by controlling the immune responses. So we've kind of had a renewed interest in orthopedic research with the advances in materials and the ability to interact with the immune system in the regenerative processes. There have really been a few, and I think each one of them is one of those aha moments, and they typically come after respected scientists or engineers say, that probably won't work. And so I remember being told that light wouldn't go through mammalian tissue, and then we used light to follow biology in living animals. And I remember the first time we looked at infectious agents replicating in living animals and tracking their pathogenesis over time. I mean, it was a real memorable moment that you can remember the moment that you saw light coming from through the tissues of an animal telling you where the pathogen was. And then we applied it to other things. I started watching tissues regenerate in vivo and watching a single stem cell give rise to the entire hematopoietic system. And we realized then that we could create synthetic organelles and put them inside cells and control the cell fates in vivo and watch that. So now, not just watching biology, but controlling it and developing new tools for magnetic control of biology. So all these things are really memorable. And there was a time when a group of optical engineers told us that our design for a miniature confocal microscope wouldn't work. And so we built it anyway and observed that we could watch single cells in the living body over time. And it was small enough to fit inside humans, and used it for diagnosis of early detection and diagnosis of cancer. So I think these moments always happen when someone says, or multiple people say, that can't be done, and then you manage to do it. You think, wow, we really did accomplish something because the dogma in the field said it wasn't possible, and we overcame that. I think it really is integrating across disciplines. So I'm trained as a virologist, and I got into imaging because we were studying viral pathogenesis by sequencing the RNA and DNA of viruses and implying from that sequence data where the virus came from. So I remember asking at the time, why can't we watch these processes in vivo? Why can't we develop the tools to watch what's going on in the living body? And so it took integrating across disciplines and then connecting investigators across these fields to build new areas of research and new areas of investigation. So I think the strength is seeing opportunity, connecting the dots, and putting programs and projects together that build on people's strengths to advance a new field. I'd say try to look in every problem that you face in the lab or in the clinic with fresh eyes, and try to take an unbiased view and dogma-free view of the possible solutions. Because it's when you look from a different perspective that you see an opportunity to solve a problem in a new way. And I think that's what's really fun about studying biology, whether it's orthopedics or another field, where you see things differently and you look at it from a fresh perspective. And I think we always have to do that and make sure that we're not getting bogged down in the dogma of the field. So I like to backpack, I like to fish, I hike, do anything where I can observe the intricacies of nature and refine my wilderness awareness skills and learn new principles of biology. A skill that I use in the lab every day and try to teach people who work with me is that with every observation you make in the lab or in life, ask the question, how did this happen and what does it teach me? And I learned this fundamental question from instructors of wilderness awareness and survival. And I think it's a real critical skill in life, just asking, how did this happen and what does it teach me? And then learning from that so you can take the next step.